Ka tangi te titi, ka tangi te kaka, ka tangi hoki a hau, ki hei Māori ora. Tēnei te pou ka eke, te pou kai a koe, ko te pou o tēnei mahi, waiaho I rautou, wānanga I rautou, kōrero I rautou. Tēnā te umu ka eke, te umu o tēnei whaihanga, ka mā takuhau tū, ka mā takuhau mahi, ka mā takuhau kōrero, ka mā moetu, ka mā moe rere, ka mā moe te whakaārahia I te ata hāpara. Kua mau kātua, mau wawa e te ata hāpara, ka mana pūkenga, ka mana wānanga, ka mau mātou, hauki e nei kawera. Nō re rā, tēnā koutou katoa. Ko te mihi tuatahi ki a tātou ronga rāua, tēnā koe. Te mihi tuarua ki a ranganui, I tū iho nei, rāua koe papa tū ānuku, E tākotu nei, mai a rāua tamariki, a rāua mokopuna, anō hoki. Kei te mihi. Ka huri aku mihi ki a rātou kua hipa atu ki te pō. Haere atu rā. Te honga māte ki te honga māti, āpiti hono tātai hono. Te honga ora ki te honga ora, āpiti hono tātai hono. Nō re rā, huki mai ki a tātai no honga ora, tēnā koutou katoa. Ko tai mai tātou ki te tautoku I te kaupapa o te whenua, o te nāhere, o te taio, o te tangata, o waku hoa mahi, anō hoki. Nō re rā, ka mihi ki te iwi kainga, nā te whātua oraki, kei te mihi. Ka mihi anō ki te iwi o te wao nui, a tiriwa, te kawarawa maki, tēnā koutou katoa. Oku mihi nunui ki a koutou, kua tai mai te pūnei, kua hui, ki te whakarongo, I te korero tuatahi, inaugural speech, o tō mātou hoa mahi, Mata Pāpore, our cherished colleague, Luitgard. Tēnā koutou katoa. Ka tauhi anō, Luitgard, kia koe. He pouhiranga tēnei. It's a very important night. E hara kaua māta ki te rongo, ki tō kōrero, ki tō mōhio, anō hoki. Kua auti, tētahi haerenga huahua, haerenga raua, haerenga takaawhio, anō hoki. Engārei, hei ahurangi koe. Tēnā koe Luitgard. Tēnā koe I tō tautoko, I tō awhina, I tō aroha, I o tauira, I o ākonga, I o hoa mahi anō hoki, tēnā koe. Mānaki whenua, mānaki tangata, haere whakamua, nō re rā, tēnā koutou katoa.
Maku koe e awhie, ki te ara tupu. Maku koe e awhie, maku rapia. Maku koe e awhie, ki te ara tupu. Maku koe e awhie.
Kia ora.
Kia ora tātou. It's my pleasure as Head of School of Environment, my name is Professor Robin Kearns, I'm Professor of Geography, to introduce Luitgard in a complementary manner to the way in which George introduced her. I'd first like to just say two things. One of a very practical nature, which is there are exits on each direction. I feel like I'm a flight attendant. And if there is any reason to suddenly need those exits, we will be guided by our trustee event coordinators in the front row there. Secondly, I would just like to convey the greetings of the Dean of Science, Professor John Hosking, who can't be here this evening, and he has vested me with the honour of introducing my colleague Luitgard. Now, the word professor has the word profess embedded within it, and that is really part of the responsibility of being promoted to professor, that we are given the opportunity to speak to the wisdom of our disciplinary background, and this is what we're going to hear something of tonight. An inaugural lecture is the symbolic first public lecture from someone promoted to the status of professor at the university. It's a tradition that goes back many centuries, and the opportunity of a inaugural lecture is one where the speaker can connect to their backstory, can connect with a general audience, and we look forward this evening to hearing a little of Luitgard's very diverse background. So just to give you a little taste of her biography, and you know, it's one of those things that even when you're ahead of school and you regularly interact with colleagues, you don't quite sometimes appreciate on a day-to-day basis the absolute diversity of their experience. It was only when I said to Luitgard today, could I please have a little thumbnail sketch of your background? I'm reading this. Wow, makes me feel very parochial, actually. Although she did whisper to me that she feels a little embarrassed about her carbon footprint, but nonetheless. Her bachelor's degree was in environmental engineering from the University of Applied Sciences in Bingen, Germany, and for that she studied the impact of logging on soil science, soil chemical and physical characteristics in the Peace River in Canada. Peace River's rather north, isn't it? And then for her MSc, which was in resources engineering, she studied sediment nutrient flow in a mangrove ecosystem in Brazil. Then for her PhD from Göttingen, Germany, she studied the subterranean carbon dynamics of a tropical forest in Costa Rica, and then going on to undertaking a postdoc that involved research in Panama, and then she came to us in 2010. And so for the last 14 years, she's been a very valuable colleague who has served as head of the environmental science discipline in our school, among numerous other service capacities, and along the way gathered together a very productive group of postgraduate students, and is clearly a very valued member of this university to be promoted to full professor. So that's enough from me. Now I'm looking forward to hearing from you. And we're going to ignore these clocks that seem to be unchanged from the last season, and they're both actually showing different times. This is going to be a timeless zone that is only going to be in accordance with perhaps my cell phone, and I might give you a wink if it's about time we had some questions, but I'm sure that you're going to be so calibrated to the next 45 minutes to leave some time for questions, and look forward to it very much. Thank you.
Thank you.
Good.
So thank you, Robin, and thank you very much, George, for the introduction. So many material classes for me to be able to understand what you have been saying. Thank you for joining me this evening. It turned out to be a quite windy night, so thank you for making it. And to take you on a journey, because there might be another surprise, because I had a career before I did my first degree. So I'll shed some light on this, but also take you on a journey introducing you to some of the ecosystems I had the fortune and the pleasure to work in, but also to learn and observe and admire the other processes and the ecosystems and what they deliver and provide us with and contributing to our well-being. Right, so let's get started. So I grew up on a small farm, and this was really end of the road. So you would, what is the saying, where only the fox and the rabbit meet and stay for the night? farms in that area have names, and this is the name of my grandmother's family, so Halter. And you will be pleased to know, so it's still a family farm. My brother inherited and is now operating it. Now, this must have been a Sunday, me all dressed up, wandering in between the orchards, because orchards were also part of the farm, because there is a farm-owned distillery, because the Kirschwasser has to come and has to be produced somewhere. So yeah, growing up, I definitely got skilled in also making Black Forest cake. But yeah, a dairy farm, a small one, especially compared to New Zealand standards. So here we were talking about 20 cows. But also what was very, very important, the forest, which is part of those farms, because yeah, it is the place where you go when you need new farm machinery. Anyway, you can imagine you're definitely exposed to nature because you've been helping on the farm, and you get to know quite a bit about weather forecasts and the ups and downs and the good and the bad years. So probably a shaping of what I ended up doing for most of my career. Now, a bit of career journey, and this is not to test your German language skills. It's just, for me, reminders, because the idea my parents had for my career was banking, administration. Even at that stage, I thought, no, that's not for me. So I went to college and trained as a biological technical assistant, which provided me with some really, really important skills. And many of my students, they still know the legacy because lab work is part of all the work we're doing in my research group. Now, how many of you know the time when Waldsterben was a topic? Oh my dear, I'm the only one. Oh, thank you, Christian. Thank you. Waldsterben, forest dieback. This was a topic, and in the midst of it, I was working for a Forest Research Institute as a technician, measuring a lot of tree cores and discs to identify the growth patterns and try to find out what's driving the decline in tree growth. So it was acid rain, air pollution, definitely one of the drivers, but highlighting already the complexity, climate played a role, silviculture, management, all these things came to the mix, and there is still some unknowns. Luckily, the forest did not die at that time. They actually grew quite well afterwards and suffer now from bark beetle dieback and other climate-related issues. I realized I need a bit of upskilling, so I started with a correspondence course in ecology. I was also working for an environmental protection office, so going from the research to more the policy side and planning side, as in a local council, in times where environmental issues just become a bit of to the forefront, and advising mayors deriving or developing policies. And here it's just an illustration, also advising and providing information for allotment gardeners to minimize their pesticide use, because that was one of the policies of the local council. Anyway, topics which are really, really complex, and I'm doing work which was usually done with people having a graduate degree, and I was paid on a technician salary. I thought now things have to change. So I'll have to get a better foundation. I'll get my first degree. And that's my first degree, environmental engineering. It was actually an engineering science degree, an amazing degree. I'm definitely, my stronger skills are towards the science side, so don't rely on my structural engineering skills. And I was especially drawn to soil science because of the passion Professor Bernhard had for the subject. So when the opportunity, not opportunity, I mean, we had to find a topic for our research thesis, I opted to do it in Wood Buffalo National Park. I have been there on an internship, was exposed to some of the challenges. This is a national park, mega clear cuts. I just could not believe it. And then they were surprised that there was issues with the regeneration of white spruce. So I was looking into whether it had to do with limitations in soil chemistry and soil physical properties. Amazing soil profiles. It's just extraordinary what you can see, what the history lays down with a mix of flooding and then the regrowth of forests. There was a danger element as well, because this is the country of black bears and wood buffalo. But also what I want to highlight here, the cabin also served as the lab, because I had to pre-process the samples to be shipped to Germany and then analyzed as part of my thesis. I ended up getting a position at State Institute for Statistics. I did not last that long, and went on to do my master thesis and did my research in mangrove forest in Brazil. As you see later on, mangroves are still part of my research. Okay. Now, the remaining part of the talk are divided into 3 parts. So first, some introduction into ecosystem functions, using soil carbon dynamics as an example, but also talking about the link between biodiversity and water cycling. And this, just to get us all onto the same page, so I'm particularly focusing on those biological physical processes influencing the transformation of carbon, nutrients in ecosystems, so the linkage between the biotic part and the abiotic part. As you can see, there is a bit of complexity involved, because there are so many factors influencing those processes. And I'm particularly focusing on those major functions like production, productivity, growth of organisms, but also then the breakdown linked to water and nutrient cycling. So this was the place where I did my PhD. I spent really a substantial amount at La Selva Biological Station. So 1,500 hectare reserve in the foothills, the Caribbean lowlands in Costa Rica, amazing forests. And what brought me to the topic, this is my PhD advisor, so thank you, Etzo, for taking on a mature PhD student, giving me the opportunity to do my PhD on this subject. So what has been found at that stage was that there is a substantial amount of carbon stored below 30 centimeters. Everyone knows that. Now, at that time and stage, and even nowadays, there's still sort of people thinking, most of the things happen in the closer to surface soil, but we should not underestimate what's stored deeper down. And even more striking is the activity which happens, the organism, the microorganisms, contributing to breakdown and transfer. So when I did my PhD, I was lucky enough not to have to dig these profiles, because they are 3 to 4 meter deep. But that did not mean taking additional soil samples, so running around with soil augers on the back and eyes on the ground because of the poisonous snakes to obtain the precious information. So just here an illustration, here the reserve, we're talking about 2 different soil types which influenced the findings. So here, one of those major processes with soil respiration, the second largest carbon flux after photosynthesis. So a flux, which is very, very important when we're looking into carbon budgets, potential implications of land use and so on. So what we have been finding, spatial differences between the soil which is derived from volcanic soil, having a higher respiration than the ones on the alluvial soil. So, driven by the amount of carbon stored in the soil, phosphorus, root activity, because what we measure as soil CO2 efflux is the sum of heterotrophic respiration plus root respiration. Now, just on a side, development of tools and techniques is definitely advancing. We call this rover. I have no idea why it was called rover, because there was nothing roving with it. We had to carry it. But nowadays, this type of equipment is a bit smaller. Now, George told me not to talk about anything climate-related. Sorry. These processes are influenced by temperature and moisture. And in this case, it's not temperature driving the amount of soil CO2 respired back into the atmosphere, it is moisture. Here with an optimum curve, so moisture enhancing the activity of microorganisms, the diffusion of gases. But then once you reach a certain point, it all goes downhill because the pores are blocked with water, less oxygen, less activity by microorganisms, but also less possibility for the gas to diffuse out. What's of even more concern is this finding. So not only looking into what's emitted back into the atmosphere, but also where is the carbon coming from, acknowledging that we have this activity in deeper layers, and then finding this extremely positive correlation between temperature and soil CO2 production. So concern, because if we have increasing temperatures, which penetrates down through the soil profile, we may see positive feedback and increase in biogenic emissions into the atmosphere. Now, carbon is only one part of the cycle. It has dominated a lot of my career, but I like water as much. And also, because carbon and the water cycle are so closely linked, we can't do the one without the other. Here talking about the green water cycle, so the water which is taken up by plants and moving through the plants, transpired, and contributing then to the rainfall. Yeah, you were wondering, how can you measure the amount of water used by plants? So we're using the so-called thermal dissipation method or Granier method, where you stuck two thermocouples into the water-conducting sapwood, and based on one probe is heated, based on temperature differentials, complex equations, you can estimate the amount of water transpired. Now, what's the context? Now, here I'll take you on a trip to Panama. I don't think many of you know this children's book, a very popular one in Germany, and expose you to biodiversity by, first of all, introducing Catherine Poitevin, who set up this first tree diversity experiment in the tropics. But also here Elise Pendel, at that time at the University of Wyoming. And now, what better to do doing a postdoc in Wyoming but working in Panama, so to avoid the winters. But yes, so she introduced me to Catherine and be able to work on the plantation. So the setup, because the question is, are more diverse plantations more productive? Basically, do the trees grow better? Because that's important for various reasons, and one of them is definitely also carbon sequestration. So the plantation was set up in monocultures, 2 and 3 species mixtures and 5 species mixtures. And as you can see here, each species has certain characteristics, right? Some lose their leaves, this is during the dry season, some develop new leaves, some are tall and skinny, and some are a bit broader, so very different in their physical characteristics. Now, what we found, so this, so as Robin said, before I moved to New Zealand, I was on a fixed-term position at the University of Göttingen, where I also did my PhD in the Department of Tropical Silviculture and Tropical Ecology. And Dirk Hölscher, the head of department, had a lot of expertise in water flux and sap flux, so a great also learning experience. And Norbert Kunert here, busy installing sap flux sensors into trees. What did we find? Here on the x-axis you have the diameter, on the y-axis you have the water use. without going into detail, what we can observe, so the mixtures, so the trees growing in mixtures, they had a higher water use compared to the ones growing in monoculture. So yes, this is basically what you want if you're looking into carbon, but do you want to have excessive water use? Because interestingly, these trees did not turn it into carbon. That's a bit of a concern, especially if you're working in a system where water is limiting. So what could have led to this? So some fundamental mechanisms to explain some of the biodiversity effects, so a complementarity effect, so lower competition because they all have their specific characteristics, they have niche complementarity, but overall on an area they have a more efficient utilization of resources. Now this could be, as I was indicating, by a change in canopy, because you can imagine here the roughness is higher, potentially wind speed is higher, which influences the amount of water transpired. So this is above ground now, but you can also observe differences in the rooting pattern. So where, depending on the species, some may have really shallow root distribution, so only be able to tap and use water from upper layers, while others may go deeper down. So growing next to each other, they may not compete for the same pool of water. Now, how do we find this out? Stable isotopes 101. So a natural occurrence, so it has nothing to do with radioisotopes, which I talk about a little bit later. But so the atoms of an element having the same number of protons and electrons, but different numbers of neutrons. So as you can see, the size of the cats differs here. The one representing oxygen 18, 8 neutrons, slightly larger one, oxygen 18, containing 10 neutrons. Difference in size has implications on the evaporation of water, for example. So this is the tool I've been using and setting up at the University of Göttingen. Here showing with these differences in their behavior, for example, when we're talking about evaporation, you have a gradient of isotopic signatures. And this you can use to identify where, for example, this one takes up its water compared to this one. Because we assume that the mix which we find in the sap flow in the xylem is the sum of the sources where those trees tap into. So quite a bit of field and lab work required to get the water out of this material and then analyze it in a mass spectrometer. And with some modeling, you can quantify where is the water coming from. Again, showing these 5 different species in their different size and shape during the dry season. Where we can see that the one which shed its leaves at the onset of the dry season relies on water from the top layer. So maybe not a surprise they lose their leaves, because this is the area where water is most prone to differences in soil water availability, because that's where evaporation happens. Now, we have another species, Tabebuia rosea, during dry season, investing a lot of energy to activate its roots in deeper layers where we have more or still water available. So they would not compete for water because they're using very different compartments of the soil. Now, some others, they go across the profile. So anyway, these complementary water use are definitely one of the possibilities to contribute to the overall enhanced water transpiration, which we found at this plantation. So I think hopefully a nice case study, again, showing the linkages, understanding the mechanisms, and applying a variety of tools to assess where the water has been taken up. Okay. I want to move on to the second part, talking about ecosystem responses to disturbance. And here I'll talk about and focus on the ecological impacts of tree diseases, which I spent quite a number of years working in that area. So forest dieback, horsebite pathogens, definitely not something new. So you may have heard about Dutch elm disease, chestnut blight, and diseases which have gone through forests, eradicated species. So what I'm focusing on is rust species. So for example, myrtle rust, a fairly recent arrival to New Zealand, and also the oomycetes, Phytophthora, where we have kauri dieback as an example. So thanks to Nick Vipera, at the time when he introduced me to the subject, he was still at Auckland Council. Introducing me to the topic, thanks to him also for his interest in not looking only into the pathogen and what it does to the tree, but also the wider perspective. So what are the implications on the ecosystem? And here we're talking about kauri forests, which it's really specific characteristics regarding their chemistry, regarding the size and their dominance in ecosystems. And through Auckland Council, we received a scholarship where Donna van der Westhuizen, a master's student, did sort of a first study looking into what is the composition of the leaf litter falling down of the trees. Is there more litter falling down because Phytophthora destroys the roots with subsequent consequences in nutrition and water uptake, loss of leaves, and so on, the dieback of the canopy and tree depths. So what he found was a really, really small scale study that they observed that less reproductive material of kauri is found in those litter traps, which is of concern because that means the next generation is not happening, because if you have less seed sources. But what we also were looking into, what are the implications on the water passing through the canopy, the so-called throughfall, which I think is often ignored, but it's definitely also an important pathway of distributing nutrients, transporting it from one location to another. And thanks to Beate Michalczyk, a collaborator at the University of Jena in Germany, who spent her sabbatical in 2015 in Auckland, we set up an experiment measuring all the different water fluxes. So here, what you can see here, the so-called throughfall collector. We developed a non-disturbing stem flow collector, so there is nothing hindering and damaging the bark. And then we also collected water underneath the organic layer. And thanks to Beate, because she analyzed all those samples and financed the analysis, what we have been finding is that there is an implication. So we see less carbon and nutrients coming into the system or passing through the canopy in situations where we have a high concentration of the pathogen. Could have various reasons. I mean, at that time, we had more questions than answers. It could be because we have less leaves, so less water being able to hit the leaves and leach nutrients and carbon off it. It could have been a change in leaf chemistry, so a multitude of potential reasons leading to this outcome. Now, funding in the space, especially if you look beyond operational and the actual implications on the host, there was funding made available as part of Saving Our Iconic Trees, Naraku Taketake, a program which just came to an end, and Marie was co-leading one of the themes as well. So I was appointed to co-lead the theme Risk Assessment and Ecosystem Impacts, together with Simon Wegner from Scion, a social scientist. So our mandate was to examine the impact of the dieback on the wider ecosystem, and also identify what's at risk and establishing baselines. And we really tried to use a holistic approach. So thank you, and George also indicated a thank you to Te Kawerau-Maki, because the Waitakere Ranges, they're still under Rahui, for granting an exemption to the Rahui for us to be able to conduct the research. But also thank you to George and Bruce Burns, because they had the foresight to set up plots in the Waitakere Ranges about over 10 years ago with no little funding. I think James was also part of the team measuring, setting up those plots, which we now have the benefit to go back and revisit and assess what happened to the growth of the trees, the host of the pathogen, the kauri, but also the neighboring species. So do we see changes in plant community composition? So just one example, because I was sort of surprised, given this is a soil and root-borne disease. Janie, thank you for being here today, doing her master's thesis, working on the amount of roots found in the soil, but also the production of roots. So what we see, and already sort of indicating potential shifts, what we see above ground also reflected below ground. So first of all, the root growth of kauri is definitely a lot less given a fairly short amount of time. So that, again, it represents also the species, potentially rather slow growing. But what we also see, the non-kauri roots, they benefit from locations where the pathogen is detected, where the trees also show symptoms of dieback, because different light regimes, less competition for space below ground, which they can use and benefit from it. So again, those ecosystem level, not only looking into the host, but the surrounding, the changes, the dynamic of systems, what's observed. Then a brief, shouldn't say brief, but the other pathogen also I've been looking at, so I'm learning a lot about pathogens and a lot about, different and new subjects. So thank you much for inviting me to be part of the successful Beyond Myrtle Rust team, looking into the impacts of myrtle rust, so an airborne disease, on here in this case, ecosystem functions. And thank you, Hoa, for, against all odds, during COVID, taking care of the plants on your balcony before we were able to move them back into the glass house. So definitely disruptive years, the same for the other program. Quite some challenges for field and lab-based scientists. But here, and also illustrating that we're working at different scales, different tools here in a glasshouse experiment, because we're definitely working on an organisms we do not want to spread and do this under ambient, under environmental conditions. And this is more the most plant physiological work I think I have done, looking into tree growth, the behavior during the cycle of the pathogen, because it has different stages, which has different implications on here the photosynthesis, which is definitely infected, but also some interesting pattern with the stomatal conductance. Again, looking into water and carbon, and then internal processes. Good. Now, part 3. How can we apply and use some of those learnings from ecosystem processes in the context of nature-based solutions to help address societal challenges? So 2 examples, looking into urban vegetation, and then also talking about blue carbon, and especially in the context of CO2 emissions and carbon sequestration. So what are nature-based solutions? A fairly new term for what others call, ranges from green infrastructure, ecosystem-based adaptation, so quite a wide range of terms. It's all related and based on processes, on natural processes. So it can be the protection, restoration, enhancement of forest landscapes. So we're doing definitely a lot in the restoration space. It could be the same or similar activities along the coast. Here we're looking into urban systems, but these nature based solutions. They're not only in the context of mitigation of CO2 emissions, it's also to improve water quality, to enhance biodiversity, so a wide range of hope or expected outcomes of these activities. So just to shed some light, being in Auckland, and how many have commented that we were almost, again, a gridlock situation tonight. And when I was walking to uni this afternoon, I thought, no, it's not Auckland going to solve this issue, because cities, the predominant emissions are transport, besides energy use and so on, and probably many of you have also estimate your personal footprint. But there's also hope, because if we can take measures and improve situations in urban systems, we can definitely contribute to a minimization of emissions globally. Now, Lena Weisert, my first PhD student here at the University of Auckland, co-supervised by Jenny Salmond. We were interested, so what is the potential of urban vegetation to mitigate local CO2 emissions? And Lena did a literature review, and for many cases is most of the literature, most of the studies have been done in the Northern Hemisphere. And one thing I definitely learned throughout my career is don't transfer knowledge which you obtain in place A to place B, because that's never going to work. In New Zealand, again, we have a particular ecosystems, we have particular tree species. So we were lacking information on what would the situation be, because what has been observed is the higher the vegetation cover, the less CO2 you see in the atmosphere. Now, how can we measure this? So a different approach, a quite complex approach, and don't ask me how long it took us to find a site where we were allowed to set up a 10-meter tall mast. Because this is what it is required, the higher the better, because you have to be at least twice the height of your average sort of building height or tree height or whatever you're going to measure, because we're using here the so-called eddy covariance method. So super high frequency measurements of wind direction and wind speed together with the measurements of CO2. So finally, we're able to find a location together with an air quality station at Howick, Botany Downs, a fairly new, recent residential development. So what did we find? So what this figure shows, so on the x-axis you have the time of the day, and on the y-axis you have the CO2 efflux, so now the CO2 in the atmosphere. Now, how to read this? So when you see a negative flux means uptake. So there is CO2 taken up out of the atmosphere here, in this case, into plant growth. So yes, we definitely see uptake, means photosynthetic activity all year round, thanks to many evergreen species we have in Auckland. Still, we had here in this case about 47% of vegetation cover, and the area is still a source. Right? Also, what we did, using in this case radiocarbon, which you can use to distinguish between anthropogenic emissions, so fossil fuel-derived emissions, which are related to transport, versus the biogenic emissions. Again, brings us back to soil respiration, to the respiration of plants. And what we found was that definitely during peak hour, you see the signal of fossil fuel coming through. But the trees grow well, but that also means their microbes do well. They break down, they respire. So biogenic emissions is also contributing to the CO2 which we're seeing in the atmosphere. Now, which then reminds us, what can we do regarding land use management to influence this how can we selection of species with the uptake, management of our green spaces. Are there ways to minimize, stimulating the breakdown and the release? Okay. Now, being in Auckland, blue carbon. Oh, thank you, Caroline, for. I don't recall when and where we met, but we have supervised quite a number of students, and we have crawled through quite some mud and mangroves across the Auckland region over the past decade or so. Blue carbon also so this is the carbon stored in oceans and in coastal vegetation. Also, promoted, and sometimes it feels like, these are all the solutions, these are the silver bullets, without really understanding or knowing enough of the system. So what we did, first of all, we knew that the area of mangroves definitely changed across Auckland and also where mangroves occur in New Zealand. But I think it was still the first study to really quantify it. So that was Suyadi's work, a PhD student using a lot of field-based work, but also remote sensing to really quantify and what is it called, area photographs to quantify the change and the increase in mangroves. What was surprising, that because we always thought, oh yeah, these are all going seawards. No, it's so-called, now I call it densification, because they did a lot of infilling, but definitely a considerable increase from 2,000 hectares in 1940 when we started quantify. We used aerial images, so the first ones are sort of available in photographs, not for aerial photographs. And to over 10,000 hectares in 2014 when Suyadi did the work. So a lot of fluctuation. But also, there was a lot of information floating around. Oh yeah, this is because of sedimentation, this is because of enhanced input. But it was also here with Suyadi's work to really pin it down, because it was just, I was astonished how closely linked it was. You have forest clearing in the catchment, and subsequently, you have higher sedimentation rates. And mangroves, they just like the conditions, so they're benefiting from modified conditions. And what is there, or are there the contribution to carbon storage? So, like in many cases, it's highly variable. Location and tree size matters. We only have one species in New Zealand, but their shape and form is quite diverse, so it's more like dwarf type stature, but we also have really tall growing mangroves. Not as tall as the ones I've seen in Brazil, and as I say, the system not as species diverse, but quite a strong zonation with distance from the estuary to inwards. It also depends whether they're growing along rivers, where you see the tallest ones, whether they're estuarine mangroves, delta mangroves. So quite a range of variability. So just along some of those estuaries which we investigated across Auckland, so quite some variation above ground. But here look at how much is stored below ground. So that's the root biomass, but also what's stored in the sediment. Just flicking back, Richard was really instrumental in developing those equations, because how do you obtain the amount of carbon stored in systems? So basically you have to harvest a few trees to get a size biomass relationship, which he did for mangrove trees, but also especially below grounds, and you can imagine how tedious and a lot of work that is to sort out roots of these clay, soft sediment, rich sediments. But that's what's required to be able to do this quantification, and then also to be able to upscale to larger scale. Yes, so that's now our contribution. We have done that also in terrestrial areas to really identify species-related carbon sequestration rates. As I say, they're all different with their growth pattern and how they relate to environmental conditions. But just want to sort of conclude that those nature-based solutions definitely have their important place when we're talking about offsetting and minimizing some of our emissions and other ecosystem benefits, because hopefully I've shed some light that don't focus on one because it's so closely linked, because if you maximize something for carbon, you have to think about what are the implications on the water or nutrient cycle. But it's also to remain realistic, right? Because there are one tool in our toolbox, but we have to really explore and understand, especially when we're talking about restoration, where there is a lot thereof. But how many restoration projects has anyone gone back and see how many trees survived and how well they have grown? Because there is a lot of studies done now calculating how much could we offset, how much could we mitigate with restoration and so on, which may end up with land to offset shortages, because then we're competing with other demands on land as well. And then also, what hopefully the examples showed, we have to know their behavior under potentially modified conditions. So may it be climatic conditions, but also when we're talking about coastal systems. Right now, our sedimentation rates largely exceeds the sea level rise, but if that changes, we have a complete modification of the space in where the trees can grow. So just to summarize, hopefully I was able to shed some light to a number of ecosystems and their related functions, the complexity, and probably a bit more work cut out for me over the next few years with all the topics which are still needs to be addressing, and to contribute to hopefully better environmental conditions in the future. So thank you, and I'm looking forward to questions. I mean, there are so many people, and it is very, very difficult. Definitely colleagues and collaborators. The technical team, because usually my requests are always a bit last minute. The environmental science team for the pragmatic approach to solve our issues and move forward with teaching and whatever we'll have to do. Colleagues and friends. And also a shout out to my youngest sister, who helped me with some of the research work, but also remains a really, really strong link to family at home and dealing with whatever aging parents and family situation throws at you. So thank you. Now.